Hey everybody, my name is William Adams and today is July 10th, 2019. So this is for my case connections, specifically Unit 2, and the cases I chose were Case 2 and Case 4. So the first case I'm going to talk about today is Case 2, and that gave us brain MRI imaging of a brain tumor. And we're going to talk about how brain tumors uptake glucose as well as somatic cells. So starting with somatic cells, the liver cells use dietary glucose in the fed state to power cells and produce glycogen when glucose is unused. This is then stored in the liver and transported to the bloodstream and supplements the levels of blood glucose to other cells in the body. This is a GLUT2 mechanism and is insulin-independent pathway. So basically, if insulin is not present in the bloodstream, the liver cells will still uptake glucose. This provides elevated levels of glycogen. The elevated levels of glycogen support blood glucose in the fasted state while in the presence of glucagon and epinephrine. In the fasted state, the liver uses lipolysis to break down adipose tissue and supply glucose to the bloodstream. Next, we're going to talk about skeletal muscles. So in the fed state, skeletal muscle uses dietary glucose to fuel cell activity. This is a GLUT4 mechanism and is insulin sensitive. This means that it will only uptake glucose when in the fed state, while insulin is present in the bloodstream. This uses the processes glycolysis to break down glucose into pyruvate, glycogen synthesis in times of excess glucose to store it, and the TCA cycle to oxidize acetyl-CoA. In the fasted state, it uses its stores of glycogen to fuel muscle activity. Muscle tissue will not release glycogen and will store until absolutely necessary for energy. Next, the brain. The brain uses GLUT1 and 3 as its mechanism for uptake of glucose. This is an insulin-independent pathway. Keep that in mind because it will come back. It only uses glycolysis, and the TCA cycle in a manner of oxidizing glucose and acetyl-CoA. The only time it will not prefer this method is in extreme starvation states where it will conserve energy until absolutely necessary for function. The brain uses hepatic glyconeogenesis and glycogenolysis to provide glucose intermediates for oxidation in the fasted states. Adipose tissue uses dietary glucose to power the cell. However, not much energy is needed to power this tissue. It's primarily used in energy storage in times of excess. This is a GLUT4 receptor and is insulin dependent. It uses glycolysis to oxidize glucose molecules and store it fatty acid synthesis as well as tag synthesis. This ultimately makes triglycerols, which will be broken down in the fasted state by the liver and skeletal muscle. So next we're going to talk about how tumor cells use their receptor. And the tumor cells use the same receptor as the cell that's infected. However, I'm just going to get into what I wrote. So we're going to compare this to somatic cells. The GLUT receptor has been identified as the rate-limiting step in glucose uptake. Without the GLUT receptor, no glucose will be uptaken into the cell. So in oncogenic cells, the GLUT receptor has been deregulated and thus intakes any glucose that's in the bloodstream, any that it can get. This has been identified through radiolabeling glucose analogs and finding that they've been concentrated more within glucose cells than within tumor cells than within somatic cells. So why is this advantageous? The elevated levels of glucose uptake will help the tumor cell in signaling for cellular division. With all the ATP a cell needs, cancerous cells are then able to divide much faster. This fuels the growth of the tumor. In brain tumors, GLUT1 and 3 are utilized. And remember how this is insulin independent. It doesn't need insulin, so it can uptake glucose in the fed and fasted states. And so what modifications may have occurred in the cancer cells to alter glucose uptake? Well, oncogenic cells modify the GLUT receptor to deregulate it. This allows for a steady intake of glucose, which allows for increase in mitosis rates. Next, I'm going to talk about case 4. And I'm going to give you a little bit of background on this case before I start. So a mother noticed that her 2-month-old daughter had been jittery for some hours after breastfeeding and not sleeping. This was all that the baby has been fed since birth. Lab results showed that the infant had an abnormally low amount of blood glucose, also known as hypoglycemia. While epinephrine was administered, there was no increase in glucose levels, but there was an increase in lactate levels in the blood. Upon further examination, the baby had an enlarged liver. I'm going to get into this a little bit now. From the known symptoms and testing, it is apparent that the baby had a deficiency in the oxidation of galactose. Now, galactose is a sugar that is formed when lactose is broken down from glucose, from lactose, and is broken down from glucose into glucose and galactose. Well, what do we know about the epinephrine that they injected? Epinephrine is associated with membrane-bound receptors. This changes the phosphorylation state of metabolic proteins. The G protein-coupled receptor cascade, which I'm going to refer to as the GPRC, receives the epinephrine signals and changes the plasma membrane conformation. This releases an alpha subunit from the membrane when GTP binds to it, and will bind further to adenyl cyclase enzyme, which is also located in the plasma membrane. For epinephrine, the specific GPRC released targets phospholipase C, which cleaves PIP2 into inositol and diacylglycerol. Diacylglycerol then activates protein kinase C, and inositol induces the release of calcium molecules from the endoplasmic reticulum. None of this really helps our understanding of the symptoms that this 2-month-old had. So what else do we know about epinephrine? Epinephrine also increases the rate of aerobic glycolysis as well as the heart rate. This activates the Cori cycle, which causes a reformation of sugar molecules to be broken down, such as lactate. Lactate is oxidized when to produce pyruvate as part of gluconeogenesis. This explains the increase in blood lactate levels when epinephrine was injected, while blood glucose levels were still lower than normal. Because pyruvate is broken down from glucose, there's no more glucose in the blood when the gluconeogenesis makes lactate. It just goes straight to pyruvate, no glucose. Gluconeogenesis is common when proper nutrients are not received over an extended period of time, and this would be expected in our patient. So why was the liver enlarged? Well, galactokinase is an enzyme found in the liver. Galactose uptake occurs here and is phosphorylated by galactokinase to galactose 1-phosphate. From there, GALT enzyme is used to make glucose 1-phosphate or glycogen further down. And so a deficiency in GALT would cause liver failure from excess galactose 1-phosphate. This accumulation is symptomatic of that of a swollen liver, as seen in this case. Galactose is toxic to the central nervous system and kidneys. So when there's excess galactose, this explains the jitteriness of the babies. And so what's my overall diagnosis for this? I would say that it was galactosemia, basically the inability to break down galactose. So now, later in life, this baby, it's vital for this baby to avoid dairy products and anything that would contain a high amount of lactose and therefore galactose. Is recommended to use alternatives such as soy-based dairy products. That was my case connections, and I hope you guys enjoyed it. And if you have any questions, please leave them in the comments.